Assalamu alaikum. This video will discuss document 6 of chapter 9, Integrating Properties of Nerve Centers. Back in document 5, you have learned about synapses, among which are neuroneuronal synapses that connect neurons with one another. Such synapses are found only in nerve centers, brain and spinal cord, because they are the part of the nervous system where the cell bodies of neurons are concentrated, knowing that nerves only contain fibers. Anyway, a neuron in a nerve center receives many afferent signals from different sensory receptors and processes these signals to elaborate an afferent signal. In this document, you will learn how does a neuron in a nerve center process afferent messages. But before we start, let's recall what happens at the level of the synapse. Upon the arrival of an action potential to the axon terminal of a presynaptic neuron, you know that the voltage-dependent calcium ion channels open, which allows the entrance of calcium ions into the synaptic terminal. Calcium ions induce the exocytosis of neurotransmitters from the synaptic vesicles into the synaptic cleft. Neurotransmitters will bind to their specific receptors on the membrane of the postsynaptic neuron. Now let's talk a little bit more about these receptors. Actually, the receptors of neurotransmitters are ion channels. Remember when we discussed the different types of ion channels present in the membrane of a neuron? Back in document 1, you've already studied about the leak ion channels and their role in creating a resting membrane potential. And in document 2, you've learned about the voltage-dependent ion channels and their role in the generation of an action potential. Well, there's one more type of ion channels. It is the chemical-dependent ion channel. It is called so because it only opens upon binding to a chemical, which is the neurotransmitter in this case. In a synapse, the receptor of a certain neurotransmitter could be a sodium, potassium, or chloride ion channel. And what ion channel is the receptor determines the response of the postsynaptic neuron. If the receptor of a neurotransmitter is a sodium ion channel, the opening of this channel upon the fixation of the neurotransmitter will cause a sodium ion inflow and thus depolarization will occur. But if this receptor is a potassium ion channel, then its opening will cause a potassium ion outflow and thus hyperpolarization will occur. Hyperpolarization will also occur if the receptor is a chloride ion channel, since chloride ions are negative ions that have the tendency to flow to the inside of the neuron. Document A of your books shows how the same postsynaptic neuron responds differently to two different presynaptic neurons releasing different neurotransmitters. In this experiment, the cell body of a motor neuron of the spinal cord is connected to an oscilloscope, while stimulating electrodes are connected to the presynaptic neurons 1 and 2. Notice that before stimulating any of the presynaptic neurons, the oscilloscope connected to the motor neuron reads a resting membrane potential. But the stimulation applied to the presynaptic fiber 1 leads to a hypopolarization or depolarization, while a similar stimulation applied on fiber 2 leads to hyperpolarization of the motor neuron. This different response of the motor neuron suggests that the receptors of the neurotransmitters released by fiber 1 must be sodium ion channels, while the receptors of the neurotransmitters released by fiber 2 must be potassium or chloride ion channels. That's why we say that the synapse between fiber 1 and motor neuron M is an excitatory synapse that leads to the generation of an excitatory postsynaptic potential, EPSP, in the motor neuron, while the synapse between the fiber 2 and the motor neuron is inhibitory and leads to the generation of an inhibitory postsynaptic potential, IPSP, in the motor neuron. Let's use this simulation to make things more clear. Here you can see one motor neuron whose cell body is located in a nerve center, either brain or spinal cord, This motor neuron is shown in yellow. You can also see in red 3 presynaptic neurons that make excitatory synapses with the motor neuron, and in blue, 1 presynaptic fiber that makes an inhibitory synapse with the motor neuron. Keep in mind that red and blue presynaptic neurons definitely release different kinds of neurotransmitters. You can see that stimulating the red fiber will generate an EPSP in the motor neuron, while stimulating the blue fiber will generate an IPSP in the motor neuron. An IPSP can never cause the firing of an action potential, of course. But what about an APSP? If the motor neuron receives several signals from an excitatory presynaptic neuron in a short period of time, the motor neuron will add up the generated EPSPs. If the summation exceeds the threshold, an action potential will be fired. This kind of summation of postsynaptic potential is known as temporal summation. Summation of postsynaptic potential will also occur in the cell body of the motor neuron if signals are received from 2 different fibers at the same time. This summation is called spatial summation. So, whether the motor neuron will generate an action potential or not depends on the result of the summation of postsynaptic potentials that occurs in its cell body. Document C of your books shows the temporal summation of the EPSPs received from the same presynaptic fiber. Notice that an action potential is only generated when the summation exceeds the threshold. Document D shows the spatial summation of postsynaptic potentials received from 2 different fibers. Notice that in this case, the summation was not able to generate an action potential since it did not exceed the threshold of depolarization. To sum up, the cell body of the motor neuron in a nerve center makes many synapses with many sensory neurons. Those synapses are not of the same type. Some are excitatory and others are inhibitory. Thus, the role of the motor neuron in nerve centers is to integrate the received afferent signals and elaborate an afferent signal after temporal spatial summation. This integration is what determines if an action potential will be generated or not, and also how many action potentials as well. This is how the nerve center integrates signals to respond adequately to a specific stimulus. Assalamu alaikum.